Hello everyone. So this is the fifth one tutorial is a in silico tutorial related with the protein structure analysis for the beginners. And here in the fourth one, I just describe a detailed information about what is the structure function relationship. So here this is suppose enterotoxin is an example and which has a high homology around 80% identical homology with colotoxin as shown. So it seems that it has a similar alpha sheet and beta alpha helix and beta sheet structure, which looks like beta sheet in purple and alpha sheet in blue cylindrical. So it has 80% cylindrical and is a highly homologous. However, in the case of remote homology, as I already described in the third one, fourth one, 422. So the TSS toxin, it has a 8.8% identical, but is a remote homologous. That is a super family, and analogous is the amino acid TNS is has a analogous in structure. So that is just your as a homework. You just go through because in the 4 tutorial I already had described a detail about what is the homologous, what are the remote homologous, what are the orthologous protein, what are the analogous protein, how they converge, how they diverge. So such type of thing. What are the paralogous protein? So I just describe, I just initiate because you also have to continue with the fourth objective, fourth tutorial. That is the fifth one. So you just go through. So these are just some examples which are by mistakenly is not shown in the fourth tutorial. So I just go through and I start from this one. Now there are some thing interesting about the protein folding. That is the, what are the folds and what. So ultimately, I just have to say protein folds. Protein fold means suppose if you have a some fold in this tutorial, I just described some other protein fold, but Rosamine, Timber, and some other folds. But suppose if have a 1 protein, 2 protein, and 10 protein, a different type of protein. Suppose the Rosamine fold is found in the first protein and fourth protein and 15 protein and 100 protein. Okay, so all then they already have protein. They already have similar fold. So that's why they already have a data bank where the fold are stored, and that just fold recognition. So if we that is a ITSR or a threading approach, I will describe in the intermediate tutorial. But ultimately now you have to just get information that there are some fold and similar type of fold are found in the different proteins. So here in this PDB structure, the upper one, they have a geometry increase the number of structure because as the age, as the year increases, the number of structure increases. But the lower one, there is a new fold. So ultimately, as the year comes, so the fold will be decreases because this fold which are already shown in the previous one. So if you have a new folds, so they have to be recognized. So ultimately, as a as the structure increases, the fold recognition is decreases. So in this example, I illustrated that as the structure or different type of structure is increased by the PDB structure, so fold is not increased as in that pattern. Fold are constant or may decreases. So this is structural classification of protein and is solved in the scope. So these are a class, family, superfamily, and fold. I already told them what is the class, what are the superfamily. So you just go through and you will get information about what are the class of the protein, what are the family. So class it group the fold according to the percentage of 3D deposition of the regular secondary structure. Family is also group protein clearly related to homology. In general, we assume they are the homologous if alignment more than 30%. I would prefer the more than a 50%, and they have a similar structure and similar function. Superfamily protein, this sequence is aligned with very few percent identical, but showing similar structure. And fold are definitely a very or we can say is a highly on fold is supposed is helix and helix. So there are some structure which are not in direction. So such type of things folds. Let's take some classical example. I would prefer that you have to just go through and Tim barrel fold and Rosamond fold. So Tim barrel fold is very famous fold, and because that I suppose Tim barrel fold is already recognized in the same structure. So as it comes in a new PDB and new PDB structure, if that Tim barrel is already recognized, so that's why we are not showing that okay Tim barrel is a new fold because Tim barrel is already in the previous fold. That's why as a age as the time increases, the fold recognition is decreases. So timber is very important because the double hydrophobic core and highly stable and found in many poly mostly in jam. It has recently proved that all of them are the same ancestor. So timber is usually hydrophobic, but inside is a hydrophilic structure. I will just go through in detail. So timbrel is a insertion, deletion, and substitution, and some protein are deleted. But ultimately, you can see that this is the timbrel is here, and it is a 90-degree orientation. Ultimately, you just go through and get information that okay, these are the beam, the middle one. Middle surface, this is a hydrophilic surface, and outer surface is hydrophobic. So usually is found in the some membrane protein. Rosamine is also found this because it is discovered by Rosamine and Duman, and it also found is very protein. So that's why, because I just go through and get some information about the what are the fold. So these are some common fold, timber and fold and Rosamine. These are just examples. So resolvent fold is suppose beta sheet and alpha helix, beta sheet and alpha helix, and say it loop in that direction. So definitely there is a drift by which the some enzymes have some active site region. So such type of things is found in resolvent fold. Yeah, this is some active site 2 binding site of the AMP and ATP. So such type of things that is a classical enzyme. The carboxypeptide. These are just proteins which have some resolvent fold. Now here I just described with some more detail about what are the all alpha, all beta, alpha plus beta, and alpha by beta with some examples. I think it would be better that have to have at least some brief information. So this is the two alpha helix. So alpha helix, if suppose if alpha helix is this thing, and if 25 degree from the first helix, so if such type of thing, so they may orientate in two different fashion. If they orientate in this fashion, so 50-degree orientation is also possible. So okay, in the next slide I will describe here. So in this case, it may have possibility that you have to create some hydrophobic core. They have a 50-degree orientation. They have a changes in the 50-degree, and if they are opposite to each other, so have they 20 degree center orientation. So such type of thing. So when you just go through a detail about the protein structure analysis, so definitely if you have a two alpha helix are close to each other, so they have a possibility to either 20-degree orientation to each other or 50-degree orientation to each other. So that's why here I just describe a brief detail, because if they are in the same orientation or superimposed to each other, so okay, some residue are just repelled to each other, and ultimately they have to create 20-degree orientation, and they also have a possibility to 50-degree orientation. Okay, now in the all alpha usually have some polar residues which are shown red in color and non-polar residues. So usually non-polar residues are create a hydrophobic core, and where these, because these are hydrophobic, so it is not on the surface, and usually they create a hydrophobic core or such type of things. So most of the some drug are have possibility to, or active site region have also possibility to be that region. So usually hydrophobic cores are the out in the inside, buried inside, however hydrophilic on the outer surface, because the protein in the cytosol which is hydrophilic. So ultimately usually hydrophilic protein on the outer surface and hydrophobic cores on the inner surface. So these are just example of globulin and protein. It also have active site region and hydrophobic core. Now the second one is all beta protein. So all beta protein, as I told, is beta, 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 such type of things. Beta, then loop, beta, then loop, beta, then loop. So it also have possibility and up and down beta barrel and beta meander. Beta meander, I also told the beta, what is beta meander, what is a Greek. So these type of things we just go through, and I have a human superfamily structure. It also hydrophobic core. Such type of structure looks like up and down beta barrels. Beta propeller is also one thing. Take suppose this is the orange one is the upper right portion. So this is a beta meander. Beta meander, as I told you, the orientation of this is suppose beta seed, then loop, then beta seed, then loop, then beta seed, then loop, then beta seed. Such type of thing, beta meander. So if two or three beta meander to each other, suppose one beta seed, then second beta seed, then third, fourth, fifth, such type of beta seed. These are beta seed one, beta meander. So this look like a beta seed, then loop, then beta seed, then loop, then beta seed, then loop is such type thing, which are shown in the upper right region in orange color. So beta meander if 2, 3, 4, 5, 6, they make a propeller-like structure, so super barrel-like structure, and they also have a hydrophobic core. So these are the possibility to be and this is also beta propeller, such type of things. There's the active site region where active site is a hydrophobic core. Greek key I also told that. So these also have some possibility there, because Greek key, as I think so, it is in shown in the third tutorial. So you just go through and have to get more information about the Greek key. But ultimately, Greek key is also beta needed similar to, but for first beta seed, then second beta seed, then third beta seed, and then fourth one is bind with the first one, such type of thing. So it have a Greek key. Such type of orientation is found, and they also have a jelly roll like structure. So they just look like in this fashion, and they have to create such type of hydrophobic core. So this type of beta, all beta, these are hemagglutinin protein, and so you have to just go through and get more information, because this is a trimer of the capsid of the virus of the flu, this is membrane fusion. So okay, this type of things is found by the beta protein, and the glucanase is also enzymes which also have such type of Greek key. These are all beta. Immunoglobulin protein also have a usually beta protein, a beta seed, all beta protein. So such type of thing, either in the case of beta member or the Greek key. Immunoglobulin, as I told you. Okay. Now the alpha beta, as I told you, alpha by beta, alpha, then beta, alpha, loop, beta, alpha, loop, beta, such type of things. So if such type of orientation is found, so it's called timberal. Timberal is alpha by beta is very good example. And highly stable structure due to the double hydrophobic cores. So such type of thing, because alpha, beta, alpha, and they also have double hydrophobic cores. So particular these upper region is the outer surface is hydrophilic and hydrophobic, and inner region is also hydrophobic and hydrophilic. So heat, because is a hydrophilic, the same thing is hydrophilic, and outer surface is hydrophilic, inner surface is hydrophobic. Then another is also another layer is also hydrophobic. So hydrophobic interaction, and is very stable protein, and that's a very common and very robust protein fold, and usually found in the membrane channel, such type of thing, and is a highly stable protein. Rosamine fold, the open C-tail structure. So flavodoxin. So these are just examples and laboratory alpha plus beta. Alpha plus beta means, as I told you, alpha, then beta, and then last is alpha. So if such type of protein is called alpha by beta, alpha plus beta, and these are some example of ribonuclease. So up to now, as I think so, you have to have some information about what are the protein structural level, and I just cover almost everything related to the protein structure, which is important for structural bioinformatics. So as I think so, you should have this information. And if you have any problem, any query, just please text me, and I will always be happy to get a good feedback, or get, I'm expecting to get a good feedback. And thank you very much. So please be in touch and just subscribe my tutorial. Thank you.